Now I will talk about the tryptophan-derived neurotransmitters, which are serotonin and melatonin. For serotonin, the precursor is tryptophan, and the enzyme for this conversion is called the tryptophan hydroxylase. It is the rate-limiting step, and it requires tetrahydrobiopterin. Serotonin is synthesized in the cytoplasm. Then it will be packaged into the vesicle, and this vesicle is called the vesicle containing vesicle monoamine transporter. And some will be after that serotonin, this vesicle carrying the serotonin will be diffused, and serotonin will be released in the synaptic cleft. Some will be reuptaken, and some will diffuse to liver and converted to 5-hydroxyl-indole acetic acid, which is excreted as you see in the right, excreted with urine. The reuptake of serotonin to the presynaptic cell is inhibited by antidepressants, and those antidepressants called selective serotonin reuptake inhibitors, or abbreviated as SSRIs, like Prozac. They will inhibit the reuptake process, and that will result in prolonged serotonin presence in the synaptic cleft, and that will result in the effect of serotonin to be left up. Melatonin is synthesized from serotonin. It is synthesized as serotonin in the pineal gland. It is involved in a sleep pattern, and it regulates the dark light circle. And as I mentioned, it is important to regulate sleep, and because of this, infants sleep a long time because they release high amounts of melatonin, while elderly people do not sleep much because they release small amounts of melatonin. So if you, yes. Now glutamate and aspartate, they are non-essential amino acids. They do not cross blood-brain barrier. They are synthesized must be synthesized in the neurons, and they are synthesized in both neurons and in glial cells. This is also to emphasize the sources of glutamate from glutamine by glutamine synthase or glutaminase that will deaminate glutamine to glutamate. Also, it is synthesized from transamination of aspartate or from reduction of alpha-ketoglutarate by glutamate dehydrogenase. And the transaminase is by glutamate oxaloacetate transaminase. As you see here, aspartate, there is a speak or a talk about if it is a neurotransmitter or not, since it is not vesicular it is not packaged into vesicles, okay, functions in minor pathways, and as you saw, it is made from oxaloacetate by transamination, and also aspartate cannot pass brain blood barrier. And this is for glycine. It is a major inhibitory neurotransmitter. It is synthesized from serine by the enzyme serine hydroxymethyltransferase through the 3-phosphoglycerate. Also, it is packaged into vesicles and the same as others, released into the cleft, and it is removed by specific receptors through glial cell or back to presynaptic cell in which it was synthesized in. Folic acid is required for glycine synthesis from serine. GABA, which is the gamma aminobutyric acid. GABA is present in very high concentration and comparable to other small neurotransmitters. It is about 1,000 times higher than other ones in the brain. GABA shunt functions to produce and conserve the supply of GABA, and its synthesis is a single-step reaction involving decarboxylation of glutamate, okay, by an enzyme called the glutamate decarboxylase. As you see, glutamine is deaminated and converted to glutamate decarboxylase to GABA, okay, then it is packaged into vesicles, and the vesicles is diffused and GABA is released. GABA is taken by presynaptic terminal and repackaged as shown here, and GABA could go into a shunt in glial cells, okay, where it's taken there and and converted to glutamate, then glutamate by glutamine synthetase converted to glutamine, and the glutamine back to presynaptic cell, okay, as glutamine, then the cycle will be repeated and gamma will be produced and goes into the cycle or into the shunt as shown. Now, for other neurotransmitters that are not amino acids from the small category, the acetylcholine. Acetylcholine, as you see, it is synthesized in the presynaptic cell from choline and acetyl-CoA. Choline could be obtained from cell membranes phosphatidyl choline or from diet, okay? The enzyme that catalyzes choline and acetyl-CoA to acetylcholine is called acetylcholine transferase, okay? And after it is being synthesized, it is packaged into vesicles, and the vesicle is diffused and acetylcholine is released. It will bind to some receptors in the postsynaptic, and it is removed by hydrolysis by acetylcholine esterase to choline and acetate. Choline will be reuptaken by the presynaptic cell, and the cycle will be started again. Histamine is synthesized from histidine by the histidine decarboxylase that requires pyridoxal phosphate. It does not penetrate blood-brain barrier. After it is synthesized, it is packaged into vesicle, and then it is inactivated after it has been released into the presynaptic cleft by histamine methyltransferase and diamine oxidase in the astrocyte, and it was found that there is no reuptake in the presynaptic neurons. And finally, it is the third category of neurotransmitters, gases, and the example here is the nitric oxide. As you see, its half-life, it is very short, 2 to 4 seconds. It is inhibited by hemoglobin and other proteins, other heme proteins. A lot of exceptions in terms if we considered the nitric oxide, if it is a neurotransmitter or not, according to the neurotransmitter definition, because there are exceptions to those definitions. But anyways, it is considered as a neurotransmitter. It is synthesized in postsynaptic neurons, not in the presynaptic as the others. And it is synthesized from glutamate. As a glutamate binds to its receptor in the postsynaptic and it enters the postsynaptic, calcium ions will enter and activate the enzyme nitric oxide synthase and convert arginine into citrulline and nitric oxide. Now, nitric oxide is released and binds, released from the postsynaptic and binds to the neighboring cells, including the presynaptic neuron across the postsynaptic that it was synthesized in. Then, as a result, it will activate guanylate cyclase, and cyclic GMP will be formed, and this second messenger cyclic GMP will activate many proteins, many target proteins. Now the question as I raised, is nitric oxide a neurotransmitter? The answer is yes, but there are exceptions. Among those exceptions, that it is not stored in vesicles as other neurotransmitters, as we see for other biogenic and amino acid neurotransmitters. It is not released by calcium-dependent exocytosis, but it diffuses from the postsynaptic neuron. Nitric oxide active process that terminate its action, there is no active process that terminates its action as other neurotransmitters. It does not interact with receptors, and it acts as retrograde messenger. That means that it comes from the postsynaptic and back to the presynaptic, and this process is called a retrograde process, and it is called a retrograde messenger. Nitric oxide is synthesized by, there are 3 enzymes, 3 nitric oxide synthases, isoform 1, in which it is found in neurons and epithelial cells, and this is activated by calcium influx. Isoform 2, it is induced by cytokines, okay, and it is found synthesized in a smooth muscle. And the isoform 3, it found in the endothelial cells or blood vessels, and it is activated by external calcium influx. All these 3 types of nitric oxide, they all require dihydrobiopterin, and requires an ADPH from the conversion of arginine to citrulline. I become very tired. Now the question that will conclude our lecture: what is the difference between neuropeptides and neurotransmitters? There are many differences in terms of activity, responses, duration of action, receptor target, if they affect gene expression or not, site of synthesis and transport, and the physical type for packaging, concentration of the action, speed of the release, the amount of calcium or the concentration of calcium that is required for the diffusion and release, also modification action, and the fate is different from neuropeptides and neurotransmitters. And this is the last slide for this lecture. I hope you understand it. If you did not understand things, please email me through emailing, through e-learning, or through the email. And thank you very much for your patience.